Hi there, this is Dr. Kingston, and in this video, we're going to be talking about the small intestine. Our objectives for this lesson are to compare the three parts of the small intestine, the duodenum, the jejunum, and the ileum, and then to describe how differences in their features allow them to fulfill different digestive roles. We're going to spend a good amount of time talking about absorption in particular, and how it relates to the surface features that we find in the intestinal lumen. So here we are. We've made it all the way down here to the small intestine now. We're back on the main alimentary canal. And now we're going to start to see how some of the secretions of the liver and the pancreas that we saw in previous videos come into play. Now, just like we saw in the stomach, there's a lot going on here in the small intestine. It has features that are going to secrete, features that are going to mix, features that are going to digest and then absorb. So let's take a closer look at all of them. So to begin with, we are going to start by dividing the intestine up into three regions. The first is the duodenum, which is separated from the stomach by the pyloric sphincter. Duodenum means 12, and it gets its name because it's approximately 12 inches long or 1 foot. Or we can call it long or we can call it short. It's pretty short compared to the other parts, but it's going to extend from the pyloric sphincter all the way down to the suspensory muscle of the duodenum, which is a little bit of a misleading name because it's not really a muscle, it's a ligament. It's also referred to as the ligament of Treitz, and what it does is to suspend the duodenum from the vertebral column. So it looks a little bit like this. It makes a little sling around here, and it has a little slip that comes up and attaches to the vertebral column. Okay, we're drawn slightly off center here, but usually this is a very midline structure. Beyond that, we have the jejunum, which is shown here in blue, and it is colored in as being the more superior part of the small intestine, but in all honesty, the small intestine packs itself in however it can. Sometimes the jejunum is sitting more superior like this, sometimes it's off to one side. You can't really tell until you grab onto it and follow its length. But this is a good drawing for reminding us that it is the second portion of the small intestine, and it's the second longest. So it's about 7 feet long, and its name means empty, which is kind of a weird name for an organ that usually has a lot of things passing through it, right? But when old-timey anatomists were naming parts, they would typically find this region to be mostly empty. I, on the other hand, am here to tell you that it is very regularly not empty, and it will make just as big of a mess as the other regions of the small intestine if you poke it with something sharp during dissection, so don't do that. Finally, the third part is going to be the pink, the ileum, which means twisted, and I think that's probably the best descriptive name of the three, yeah? The ileum is roughly 11 feet long, so 7-11 is a good way to remember these two, their lengths. And that ileum then is going to extend all the way down to the right side of the abdomen at the ileocecal junction. Now, on the outside, the regions of the small intestine look pretty darn similar. There really isn't a good way to tell them apart visually without looking at their blood vessels or taking a look at their internal features. But in order to even do that, we've got to get familiar with what their internal features are to begin with. So let's start on the inside here. We're going to start at the deepest part of the intestine, at the mucosal layer. Now, just like any mucosal layer, so we are in pink here, it's got its 3 sublayers. So it's got this muscle down here that changes the shape of the mucosa. It's got this middle connective tissue layer here. Okay, loose connective tissue. And then finally, it's got an epithelial layer on the surface to line the lumen. Now, the defining feature of the small intestinal mucosa are these finger-like projections here called intestinal villi. Their purpose is to expand the surface area of the mucosa so that there is room for more epithelial cells. You can see these drawn on here. These are absorptive cells, goblet cells, and enteroendocrine cells that are found on the surface of the villi here. Deep to that epithelial layer, then, the loose connective tissue here, or areolar connective tissue, is going to provide some support scaffolding. So just like in the stomach, the loose connective tissue layer here is called the lamina propria. And again, this is where we are going to find our capillary beds and lymphatic vessels. So specifically, what we see here are these long lymph stalks that stick up. They're called lacteals. They stick up into the villus, and then they are surrounded on all sides here by the capillary beds. All right, so that's a villus. Villi are the bits that project up, but you might also notice here that we have a part that sticks down into the lamina propria as well. And these are the glands for the small intestine. Okay, just like we had gastric pits in the stomach, here we're going to have intestinal crypts. And these are lined by secretory cells. So absorptive cells again, goblet cells, and Paneth cells. Now, the villi and the crypts of the intestinal mucosa are very, very small. You need a microscope to see them clearly. That's why I'm showing you illustrated pictures of them. But one feature that you can see clearly with your naked eye are the many folds here that the mucosa layer is arranged into. And just like the villi, these folds serve to expand the surface area of the mucosa, but they do it in more of a macro sense where they're adding inches and feet instead of micrometers to that surface area. So these folds tend to encircle the entirety of the lumen, so they stretch all the way around. They take the name of plica circularis, or circular folds. And having them arranged in a circle like this not only expands the surface area of the mucosa, but it also forces the chyme passing through here to kind of spiral through the lumen, and it catches some of that chyme as it's passing through, so it's really maximizing the surface area for contact, direct contact with that chyme, which in turn allows the lining to absorb more nutrients. All right, so now that we've got everything named, let's look at what's going on for secretion. Now, interestingly, despite its massive length, the small intestine really doesn't secrete much in the way of its own digestive fluids. So as we've seen, it gets a lot of help from the liver and pancreas. But what it does make for itself is a mixture called intestinal juice. And that is a mixture of mucus that's secreted by these orange goblet cells here, some digestive enzymes that come from the absorptive cells, and lysozyme, which is a bacteria killer that is secreted by these yellow Paneth cells down here. The other thing that is secreted is going to be hormones. So we've got these blue enteroendocrine cells that are kind of dispersed all the way through the intestinal lining. These are going to secrete gastric inhibitory polypeptide, or GIP, and that is going to stimulate insulin secretion in the pancreas. They're also going to secrete cholecystokinin and secretin, which are going to tell the pancreas to secrete some juice and for the gallbladder to contract. So we covered both of these in the endocrine section, so I'm just going to leave them here as kind of a quick hit. Mixing in propulsion is once again going to be carried out by the muscularis layer. The muscularis in the small intestine is pretty bog standard. 2 layers. We've got longitudinal fibers superficially, and then circular fibers sitting deep to those. These are going to coordinate their contractions to move the chyme traveling through here in a couple of different ways. So firstly, just like we saw with the stomach turning, we have a motion here whose entire purpose is to shake up the contents of the small intestine and to mix them around. This is called segmentation, and it consists of circular fibers contracting in alternating groups. So here in this top image here, we've got A representing the start of this process, where we've got one specific group of circular fibers that contract at the same time, and that separates the contents here into blue and yellow. All right, in the next step, B here, we have an alternating set of fibers that's going to contract now, and that mushes the contents from these original groups together, starts to mix them up, and then those first fibers contract again, and we end up with some nicely mixed green sludge traveling through our intestine. Okay, so that's how we have some mixing going on in the intestine. The second motion is just good old peristalsis. And again, we've got circular fibers that contract superior to the chyme or proximal to the chyme that it's going to move through. And that forces that to move distally. Some more contract, moves distally again, and then more contract above that. So now that all these juices are getting mixed up with the chyme passing through, let's take a look at what they're doing. Now, we've said many times now, most of the chemicals that digest food in the small intestine come from outside. So it is importing bile from the liver, okay, and it's importing pancreatic juice from the pancreas. But it does secrete its own intestinal juice, and that juice includes a number of digestive enzymes. Okay, so together we've got bile coming down from the liver through the bile tract and gallbladder through the cystic duct, bile duct here. Bile duct meets up with our main pancreatic duct here to form the hepatopancreatic ampulla, which enters into the major duodenal papilla and into the duodenum itself. Meanwhile, our duodenal lining here and the small intestinal lining in general is creating those digestive enzymes that will continue to help break things down. And together, all three of these are going to complete the digestion of carbohydrates, proteins, and lipids that are passing through the digestive system. And once all of those things have been appropriately broken down, they can now be absorbed into the intestinal wall, and from there, the bloodstream. Now, absorption happens through absorptive cells, so that's nice, right? The absorbed nutrients are going to pass across the cell's membrane. So we've got nutrients out here. They're going to pass in just like that. And now we have to look over here. So they've come in, and now they're going to be absorbed into these capillary beds here, and they'll be taken away by our veins. These are all portal tributary veins, remember. And those will join to create bigger and bigger vessels until they reach the portal vein, right? Okay, now this is where having all of the surface area from the intestinal villi and the intestinal microvilli here really comes into play. So more plica circularis is going to create more surface area for the mucosa. More surface area means more room for these little absorptive cells, and more absorptive cells mean more of their little microvilli. So we're building surface area up on multiple layers. Now, instead of doing a regular practice question for this lesson, I want to walk through a two-step question here with you. And to do that, I want you to take a look here at what structures are found predominantly in the different regions of the small intestines. And I want you to think about how that might affect some of the digestive processes that take place there. So to begin with, in the duodenum, we find many well-developed plica circularis. And we have that major duodenal papilla sitting here as well, where the hepatopancreatic duct enters in. Right? In the jejunum, we have, again, many plica circularis covering the walls here. The walls are pretty thick, and the lumen here is quite wide. Over in the ileum, we find fewer and shallower plica circularis, and these are covering thinner walls, and they are surrounding a much more narrow lumen. So how might those characteristics of each here be related to what they do for digestion? Where would you expect to find the most chemical digestive taking place? What about absorption? Okay, so take a few seconds here and think about it. Now, the gradients for absorption and digestion look just like this. Both are going to happen across the entirety of the small intestine, but there are definitely hot spots here for each. So chemical digestion is occurring mostly down here in the duodenum, and that is because that's where that main pancreatic or the major duodenal papilla is located. Okay, so we have most of our digestive juices coming in right here. So they start digesting things right here. Chyme here then gets really thoroughly mixed with those juices through segmentation, and then it's forced to spiral around these plica circularies as well. And that's going to kickstart the entire process so that by the time that chyme reaches the jejunum, a lot of that chemical digestion is already done. So once we've got chemical digestion finished, we can really get to the business of absorbing things. And that's just what we see here, okay? So we see absorption really pick up in the jejunum. And again, we've got lots of plica circularies and absorptive cells for this. So that helps out very well, okay? Not only do we have many plica circularies, we've got very thick walls and a very wide lumen for things to pass through here. Okay, so we do a lot of business here in the jejunum. Once we get down to the ileum then, we see we have that much narrower lumen, much thinner walls, and much shallower and fewer plica circularies. And this is not because the ileum isn't good at its job. It's just that most of its job is already done by the time it gets there, okay? So we want to think of the ileum as kind of catching the stragglers that haven't been fully digested and fully absorbed by the time it reaches there. One thing that I want to point out is that these characteristics of the jejunum and the ileum, I keep saying the jejunum has lots of plica circularies, the ileum has few. These are not like you hit a landmark and suddenly there are fewer plica circularies and thinner walls. These change as a gradient. So the jejunum and the ileum are going to grade into one another. Plica circularies in particular, they become fewer and fewer and they get shallower and shallower the more distally you travel in the small intestine. The walls become thinner and then the lumen gradually becomes narrower. But in general, it is good to be able to compare some of the characteristics of the jejunum and the ileum, both to help you find your way around anatomically and to help you sort out their different functions. So here, I put together a quick comparison for you. The jejunum here, 7 to 9 feet long, lots of plica circularies and a relatively wide, about 3 centimeters lumen, thicker walls, and the arteries that supply it. So the reason I'm including this is because surgeons will often shine a flashlight on these to see which part of the small intestine they're looking at. They have few arcades, so arcades are these loopy parts, and they have long vasa recta, or these branches that come down and specifically serve the jejunum walls. The ileum then is going to have fewer plica circularies. It is between 11 and 12 feet long. Its lumen is decidedly narrower, only about 2 centimeters wide, and its walls are also thinner. The arteries coming here are going to have lots of little arcades and shorter comparative vasa recta. All right, and here is your overview slide. Again, the small intestine is primarily part of the midgut, but it does have a small contribution from the foregut as well. So that would be the first parts of our duodenum. So make sure you understand which portions are supplied by which arteries and which nerves, okay? So while you're studying, make sure you're practicing knowing where that cutoff is. All right, and that brings us to the end. So I will see you in the next video.